Welcome to the Personalized Medicine Podcast. This is the place where scientists, clinicians, and entrepreneurs discuss the progress of this rapidly developing field. I am your host, Oleksandr Yagensky. Let's start. Three, two, one, and we are live. Welcome to the next episode of the Personalized Medicine Podcast. On this episode, we dive deep into a new group of biomarkers that we have not covered yet on this show. Today, we will discuss small molecule metabolites and how they can be utilized to evaluate patient-specific response to therapeutics. I am very happy to welcome today on our podcast a scientist and an entrepreneur who is shaping the future of this field, Dr. Liz O'Day. Liz obtained her master's degree from the University of Cambridge, where she studied biophysical properties of alpha-synuclein, a key protein involved in Parkinson's disease. She then moved to Harvard University to do her PhD on RNA structures and metabolites in tumor-initiating cells. In 2014, Liz founded Olaris, a biotech company that leverages metabolomics and machine learning to determine optimal personalized treatments for specific diseases. For the last 4 years, Liz also has been a co-chair of Global Future Council on Biotechnologies at World Economic Forum and a member of advisory board for women in the enterprise of science and technology community. Liz, for me, you are an embodiment of a successful research-driven entrepreneur. Thank you so much for accepting an invitation and welcome to the show.
Thank you so much for having me and for that super generous introduction. I hope one day to sort of be able to live up to what you just said there, but thank you. I've been listening to your podcast, and the group of scientists and business leaders that you have curated is truly amazing, and honestly quite honored to join this crowd. So thank you.
Great. The pleasure is ours, and I'm really, really happy that we finally found time to sit together and talk. So I would like to start with Olaris, and it is such an exciting company on really many levels. And I would like to learn about the story behind this company. So what have motivated you to start it in the first place, and why actually metabolomics?
Yeah, great questions. Well, I would say that the mission of the company actually got started when I was in kindergarten, and my older brother was in first grade. He had been, losing weight, not eating, pretty tired, not, typical for your average 7-year-old. And after bringing him to see our primary care physician, a couple of times, running different tests, I distinctly remember the day that my parents bundled us up and dropped my younger brother and I off at my grandmother's house. We called her Nani. And they brought my older brother, Rob, to Boston's Children's Hospital, where he would be, at that point, formally diagnosed with a childhood cancer called neuroblastoma. It's a rare form of cancer of the adrenal gland. And the way I thought about it then, and it's actually pretty much the way I still think about it now, is if you took a wet sponge and put a stone against it, it would cause sort of like liquid to leak out of the sponge. And basically that same thing was happening to my brother. A tumor was pressing against his adrenal gland, forcing out, all these different types of molecules, metabolites, and hormones, and completely like messing up his body. And now I'll fast forward and just say, my brother is a survivor. He is, enjoying living out his 30s on Nantucket, working as a carpenter and as a contractor. I often joke that he made way better life choices than I did because he wakes up every day on the beach and gets to go build houses where typically I'm locked away in a lab or a library. But from that day that he was diagnosed, he actually spent, and I guess in large part our family did too, the next 2 years completely living in Children's Hospital. And, even though my parents, the doctors, the nurses, everyone definitely tried to shield me and my younger brother from it, even as a 6-year-old, I saw how serious this was. And I wanted my brother to be better. My brothers and I are super tight. We would build forts, play superhero games, and perhaps it's really selfish, but I just wanted my big brother back, and I wanted the tubes out of his arms. I wanted the IVs. I wanted all the monitors gone. And I would beg the doctors literally to, just fix him, is what I would say. And even in like the medical records, there are notes that like his little sister is really not coming to terms with the seriousness of his condition, because, I just wanted him better. And when I would ask them like, well, what can we do? They would tell me they were doing everything that they can. And all I could do was like hope and pray that the medicine worked. And, good Lord, I did. I did both of those things. But quite honestly, I found that pretty dissatisfying. I believed there had to be a way to know how to help him the best. I thought that we have to move past this sort of guesswork and find a way, for my brother in particular, to make him better. And it was this experience that actually motivated me to be a scientist, to become a cancer researcher. And truly, this has been my goal, ever since then, is that I've pretty much dedicated, I'd say, the majority of my waking hours trying to learn about cancer, acquire sort of the necessary research skills, and to develop technology that could truly try to solve, that problem that my brother faced all those years ago in Children's Hospital. So that's where sort of the idea, or the motivation for Olaris came together, way before I even, had an inclination about starting a company or anything like that. And then, from the science side, as you kind of alluded to in my introduction, I've been in a lab the majority of my life. And even as a kid, I was into cancer. If you can be into cancer or science, I spent lots of time in the library and reading things, and as soon as I went to college, pretty much on my first day at campus, I started working in a research lab. I was originally trained as a synthetic or organic chemist to make small molecule inhibitors. That was at Boston College. And then, as you said, I jumped across the pond to learn a little bit more biophysics. And that really taught me the importance of understanding how things kind of come together on a molecular level. And then when I went back to Boston to do my PhD at Harvard, I continued to sort of embrace this idea of trying to learn everything. And I actually pursued a joint thesis that put me in two labs. One was a biophysical lab led by Gerhard Wagner doing NMR spectroscopy, and the other was cancer biology led by Judy Lieberman. And, this sort of dual PhD, let me get access and become privy to all these cool different techniques and all these cool different things that just kind of helped me build up my arsenal of, as I was thinking about how can we tackle, cancer and drug treatment problems. And the aha moment I can remember was in, maybe my fourth year of my PhD, when I was reading a bunch of papers, and I remember them quite well. They were by Matt Vanderheiden at MIT and Luke Cantley and Craig Thompson about cancer has a sweet tooth. And it was this idea that cancer is addicted to glucose. And we've known this forever. It's the Warburg effect. Cancers, uptake a lot of glucose, but not to make energy. They feed other pathways. But that even from, the time of Otto Warburg up to now, we as scientists really hadn't figured out why cancer, eats all that glucose and where does it go. And it just kind of clicked in my head, all the training I had from a synthetic chemist, from a protein biochemist, and then to an NMR spectroscopist, that we could design a technology that could help us figure that out. And in turn, that would probably help lead to these, developing diagnostic tools to stratify patients. And so I would say, so from then on, it was on, I would say, where I spent, the rest of my time at Harvard and then post then sort of developing this technology that would allow us to probe metabolism and then use that information to ask interesting questions, to figure out what's the difference between cancer versus healthy, or drug responsive versus drug resistant, and so on and so forth. And again, I had zero intentions in my entire life of starting a company. It was only the fact that I felt in my bones that this technology, could really help real patients. And I didn't want it to just, be stuck in an academic setting. I wanted to get it out to people that truly, without probably knowing what I was doing, 6 years ago now, decided to start the company directly as a spin out of my PhD. And I'm happy to say we're still here today.
Yeah, that's a fantastic story and a very intense and personal story as well. I think it's really important when founders care deeply about things that they do with their companies. And I think you couldn't have been more motivated having all of those experiences starting in your childhood to found specifically this company. And what I would like to ask you next is actually to give us a little bit more of specifics on what actually you are doing at Olaris. I know that you are studying so-called biomarkers of response. Perhaps you can tell our audience what those biomarkers are and how can we use them to stratify different cancer patients?
Yes, great question. So biomarkers of response, or we call them BORs, are biological molecules. So something your body made or is within you. So this you can think of like DNA, RNA, proteins, metabolites are examples. And these biomarkers correlate with a particular phenotype or outcome. In this case, Olaris, we measure small molecule metabolites. So this is all the molecules that swim around in you and I that provide both the energy and biomass for life to exist. So, you know those people that can eat whatever they want and they stay thin? Is that you?
It definitely wasn't me in the childhood. Probably now a little bit, but I think it's starting to change again.
Well, the pause there feels like the answer is yes, and like you and I, we'll still find a way to be friends. But so people like you or people like that, you know, everybody has a different metabolism. So like the way that you take in food and you break it down and you synthesize it into different molecules, that is specific to you, and the way that I do it is sort of specific to me. And we will make and break endogenous molecules, things that our body sort of needs, but because we are humans and interact with the environment, all the other stuff that like is expressed in the world or we come in contact with also gets captured in your metabolome. And so Olaris develops or has, now a patent pending metabolite profiling platform that lets us sort of detect and quantify all of those particular metabolite biomarkers that then we feed into machine learning algorithms to find patterns that are associated, with particular responses. We're not the only company out there, trying to identify biomarkers for anything. There's lots of them out there, and people can look across, different classes of molecules. There's genetic biomarkers. Like if you think about Huntington's disease, there's a series of repeats in one particular gene. If you have it there, it's a pretty strong indication that you're gonna, get the disease. But there are other biomarkers that, are not just like a single agent that you're measuring, but are actually more like panels. So there's a pretty well-known test now in precision medicine called the Oncotype DX test, which is made by Genomic Health. And they measure, 21 genes, and they use sort of the expression of these 21 genes to predict recurrence for early stage breast cancer patients. And, there are probably, other biomarkers that are going to develop in the future that even kind of cross classes where, so like Genomic Health's got some genetic ones, Olaris has some metabolite ones. In the future, I suspect we'll even see sort of multi-omic ones where it might be some genetic component coupled with, a metabolite or an epigenetic or a protein signature as well.
Perfect. Yeah, that sounds really exciting and promising. And I guess the combination of that multi-omics techniques can give us even better view on the diagnostic kind of background of specific patient, specific person.
Right. Like I think collectively that helps us sort of understand the system, right? Sometimes if you're just measuring, genomics, you're looking at a moment in time and particularly like a tumor, but things are changing, right? So having additional biomarkers that can monitor those changes, I think are really important. And again, influence with the environment. We don't live in a static world. So we're constantly inundated with things, and understanding how all of that comes together from, microbiome to like what we eat, what we drink, all of that is going to be really important to figure out what's going on.
Exactly. And you mentioned that the human body and essentially our whole organism is very dynamic. How dynamic is our metabolome, and what of those dynamic changes can we capture with modern techniques?
Yeah, so metabolism is thought to be sort of one of the first sort of readouts of change, right? So, when something don't know, goes wrong, or there's an altered pathway, that metabolites are, one of the first responders, let's call them, to sort of set off a signal or a hallmark that like something has changed. So your metabolites change on the daily, on the minute, right? If you've eaten, if you've exercised, if you've, haven't slept in a while, like all of that kind of gets reflected in your metabolome. That's what makes them, such a powerful biomarker for phenotype, or for determining different sort of classes of phenotype. It also makes it a bit of a challenge, and why you need to be very sort of rigorous in how you set up metabolite-based biomarker profiling, so that none of those other potential factors, like did you just eat a cheeseburger, influence our studies to pull out, diagnostics. But really we find metabolite biomarkers that are associated with, again, like drug responsiveness or not. And so the setup of the experiment and the pulling out of these biomarkers is incredibly crucial, and has to be done sort of very carefully in order to pull out sort of meaningful data. And Olaris's approach relies heavily on two major sort of analytical platforms, NMR, nuclear magnetic resonance spectroscopy, and mass spectrometry. And the reason why we use both of these platforms is because metabolites are quite diverse. So there are metabolites that are really greasy, like hormones and lipids. And then there are metabolites that are polar, like maybe acids and things like that, nucleic acids or amino acids, or ATP. And there is no single analytical platform that can detect all of those metabolites in one go. Not to mention, metabolites can range across like 12 orders of magnitude in concentration. You can have teeny tiny things, maybe in the femtomolar range, and things like glucose, which are consistently in the millimolar range. So you need to kind of combine different platforms that can let you get the broadest coverage of the metabolome, but also the most rigorous. And that comes again by setting up your experiments right, say setting up the clinical data as well, and sort of capturing the sweet spot of biological data and metadata so that you can pull out sort of interesting signatures. And that's, again, where kind of the machine learning part of what Olaris does comes into play.
Perfect. That sounds really exciting. And I guess these two groups of technologies that have seen a lot of development in recent years, NMR and mass spectrometry, are receiving more and more attention today, especially in personalized medicine field. Perhaps you can elaborate a little bit more on the specific use cases where you would prefer to use NMR over mass spectrometry, or vice versa, to assess specific metabolites.
So NMR and mass spec are often pitted against each other, but truly they're quite complementary techniques. So there are certain things that you can easier detect, on NMR that you simply would not be able to see on mass spec, and vice versa. So we don't really have an either or sort of viewpoint, but a both. NMR really good at like aqueous metabolites. You're considered what's called like central metabolism, so amino acids, glycolytic products, and things like that. Mass spec, you can design specific methods to pull out, particular, let's say, like lipid classes or different molecules that might not be as easily identifiable by mass spec or by NMR. So I guess, I don't know if that's maybe a cop-out of an answer, but I think that they're both helpful. It's just, combining them to get sort of the broader viewpoint of the metabolome is at least our approach.
Right, kind of like you need different types of screwdrivers in your toolbox to do the job.
Yeah, that's an excellent analogy. I like that.
Okay, great. And you mentioned also that you extensively use machine learning in your approach as well. I think machine learning kind of penetrated pretty much every field of technology, every field of science, but specifically how machine learning helps you to analyze those biomarkers of response in human samples.
Yeah, I agree with you. I think, AI and machine learning is everywhere. And I am obviously a fan of it. I think that both of these technologies can help make lots of the way we live better. And I think in particular, healthcare will become more efficient as more AI and ML get integrated into sort of normal care. At Olaris, what we do is we use machine learning to find patterns of metabolites that can help us identify people who did well on a particular drug versus those that did not do well. The simplest way to sort of think about it is like facial recognition. So, if you and I took a photo and threw it up on Facebook, Facebook would suggest, oh, is that Liz O'Day over there? And be able to tell us apart. And it's not because like you have a nose and I don't. It's that there's sort of different ratios, between eyes, nose, mouth, whatever, that let these algorithms sort of tell us apart. And so Olaris does sort of something like that, where we try to find like ratios of metabolites. Again, it could be endogenous, microbiome, xenobiotic, that let us build a signature that can differentiate drug responders versus drug non-responders. But what makes us a little bit different is that we go one step further. So I don't really believe in black box machine learning and AI, where you just like run the algorithm and it gives you some model, but you don't understand, what's going on underneath it. I think especially in healthcare, we need to kind of go that next level and figure out like, what are the features? Like why, is the algorithm classifying these people, into these two different groups? And so that's what Olaris really spends a lot of time doing, is like digging into the algorithm. So we will run, there's millions of machine learning algorithms out there, and we will run as many algorithms that make sense for a particular data set. We won't look for the algorithm that gives us the best answer. We look for the features or the metabolites that are most important in distinguishing our two groups of patients, for example. And then once we identify those metabolites, we then build our own customary algorithm that takes into effect kind of the ratios of these metabolites to try to differentiate the two groups. But we always go back to the biology and say like, okay, if these metabolites are upregulated, in people who are resistant to a particular drug, one, does that make sense? And like two, what does that teach us? Does that teach us something about, the resistance mechanisms? Does that teach us or give us ideas about potential new treatments or co-treatments? Or how can we use this information to kind of move our understanding of the disease or our ability to treat the disease further?
Understood. Yeah, it makes perfect sense. And as you mentioned, it's more than just a black box. You really need to understand what is happening inside of it to be able to interpret the data and actually make most use out of them. And from what I understand, essentially you are building like Facebook for metabolites.
That's what we're trying. Are you a comic book fan?
Not that much. I had a few, but no. My childhood was I was rather like a fan of TV shows, but
okay.
Not comic books.
Well, if you read the X-Men series, Professor X and Magneto, they're nemesises, but one time they were friends, and they built this giant computer that they called Cerebro that let them like identify and connect with all different mutants across the world. And Olaris is building a Cerebro database for metabolites that like let us identify, fingerprints of metabolites or expression levels or signatures of metabolites that correlate with all kinds of clinical outcomes, response to Herceptin, response to Ibrance, even whether or not you're likely to have Parkinson's disease is another area that we're working on, and things like that, and building kind of this big database of all these metabolite signatures.
Perfect. Yeah, I think that explains it perfectly, and we'll make sure to put the link to the X-Men comic book to our show notes with all the illustrations for Magneto and Professor X collaboration. I think that would be great.
We are doing this show for you, and your feedback is very important for us. So if you have any suggestions or comments, would like us to cover a specific topic or recommend a guest, please write us an email to team@pmedcast.com. Or you can reach out to us on LinkedIn, Twitter, or Facebook. Just type in Personalized Medicine Podcast and you will find us there. To download the show notes for this episode, visit our website pmedcast.com. It's pmedcast.com. The show notes include guest bios, links to their most notable work, and recommendations for additional reads on the topic of the episode. Make sure to check them out. And don't miss the next episode. Subscribe to our podcast on your favorite podcasting platform. Give us a rating and leave a comment. It will help us make this show better. And now, let's get back to the interview.
One more topic that I wanted to discuss with you on a slightly more serious note would be the actual application cases for your technology. I know that recently you entered a collaboration to study the response of patients to specific monoclonal antibody treatment, trastuzumab. This is the antibody medication against HER2-positive breast cancer. Can you tell us a little bit more about this study and how can you leverage your knowledge of metabolites and metabolic biomarkers to stratify those patients?
Yeah, so this is one of the studies that we're working on, and happy to chat more about some of the other areas we're in as well. But we published and presented this work at the San Antonio Breast Cancer Symposium in 2019 when conferences and traveling were still a thing, and hopefully we'll get back there as well. But what we were super excited about this study is that, we did it in collaboration with Penn State Cancer Center with Dr. Alan Lipton to try to untangle this trastuzumab responder, non-responder problem. So trastuzumab is the generic name for Herceptin, one of the first examples of a precision medicine drug that it's a monoclonal antibody that will bind to HER2, this growth factor receptor that is overexpressed in about 20% of breast cancers. And, this antibody will bind there and basically shut down signaling in this pathway, and has been incredible in transforming kind of the prognosis for the HER2 landscape. Herceptin's, now known as trastuzumab, or whichever way you call it, was the first one to market, but there are lots of anti-HER2 drugs out there. There's pertuzumab, lapatinib, there's all kinds of other ones. And when and if somebody gets diagnosed with HER2 breast cancer, and this goes for many diseases as well, like patients are walked through different steps, right? So they'll probably start on trastuzumab, they'll see how it goes, and then if they progress, they'll go on to some other therapy, and then if they progress, they'll go on to something else, and it's this sort of trial and error system. And unfortunately, that's just not effective, because there are certain people who will never benefit from that first-line treatment, so you are wasting precious time as well as money on a drug that's not going to benefit someone. And then, even in the future, you're not optimizing the drug for where the tumor has grown and developed, or you're not optimizing the treatment for where the tumor has gone. You're just optimizing it, based on average outcomes. And so this study was small. It was with 26 patients who had metastatic HER2 breast cancer, and we got pre-treatment serum, so meaning before they ever started treatment. We ran it through our platform, and we identified a panel of metabolites. There were 3 that when we ran all of our machine learning algorithms, we whittled it down to our favorite 3, and then we made this scoring system that let us predict with 96% area under the curve, so incredibly high accuracy, which one of those women, would have a good response, and meaning they had a progression-free survival of greater than 140 days, versus those that did not, those that despite taking this drug that was probably told to them that was going to sort of potentially save their lives, it didn't, right? And again, it's not, I love Herceptin. It's one of my favorite drugs out there, but there's room to optimize who should get it and when.
Absolutely. And I think this is actually the personalized medicine in action, when you really determine specifically for each individual patient who is likely to respond and who will rather not. And as you said, that can save precious time for the patient, also probably spare them from possible adverse effects of the drug, and also save money for the patient and for the entire healthcare system. So this is a very important mission that you pursue.
Well, you just said, that's often what I kind of say our three-part aim is to do exactly what you just mentioned, right? To improve outcomes, to, reduce unnecessary exposure to toxic side effects, and also remove some of this incredible waste that we have in the healthcare system on ineffective treatments.
Perfect. And I actually want now to shift with you a little bit more to the slightly political and maybe organizational side of things. It is, of course, great to have very advanced technology at hand, but especially in healthcare, it's sometimes really hard to convince all the stakeholders that this is something that they should use. And in healthcare, there are obviously so many stakeholders. You have doctors, you have patients, you have payers, and all the other parties. How do you bring them to the same table, and how do you convince that what you do actually makes sense and can benefit all of them?
Yeah, so healthcare, especially here in the US, is really messed up, right? Where there's all these different stakeholders and they're all incentivized by very different things. And so I think that has led to a lot of, the inefficiencies that we currently experience here. The great news is that biomarkers of response are something that can align all stakeholders, right? So patients want to get better. They want the drugs that will help them. Physicians want to have their patients get better, to deliver the optimal treatments. Payers or insurance companies, they want to pay for and reimburse the treatments and the strategies that are actually going to improve their members' outcomes. And then biopharma wants their drugs to work. Like everybody that went into becoming a scientist went into it, because they wanted to use their scientific knowledge to make medicines that could sort of help people. And so biomarkers of response are this beautiful thing that actually can help us all sort of work together to have, a better healthcare system and hopefully even just better health in one day. And so actually, we feel very fortunate that there is tremendous support from all of these stakeholders for what Olaris is doing. I've spoken on podcasts for different patient advocacy groups, or even just tried to educate, people about how our tech works. We make it a big part of what we do is to collaborate with physicians, because, physicians are constantly inundated with all this cool new sexy tech, and, understanding what's real, what's going to be meaningful in their clinical practice is quite honestly very hard. So as Olaris develops our technology, we work with clinicians. They are true partners in us in developing our technology. And then, payers and biopharma, we are actively engaged with them as well to make sure that, they understand how our tech will help them, meet their incentives as well. And so I actually think biomarkers are one of those rare things that can bring people together in a healthcare system. But you always need the data to showcase that, right? So for us, we're doing all these scientific studies, to showcase how our biomarkers of response can improve outcomes and again, hit those goals that we talked about across different areas. We're working in breast cancer, we're working in Parkinson's disease, we're working in gastrointestinal stromal disease. We've gone from these small pilot studies, we just talked about for Herceptin, to now larger validation studies with upwards of 300, 500 patient samples there that we're working through. And that should give us sort of the scientific or the analytical piece to know that our technology works. But we're also figuring out the other half of it, the economics, right? So we're doing all these what are called health economic outcome research studies, or HUR studies, to demonstrate the waste that ineffective treatment has, not only in outcomes, which is in of itself terrible, but these drugs often cost, hundreds of thousands of dollars for a year of treatment. And some percentage of that is just thrown out the door on ineffective, on patients that it's never going to benefit. And then you couple that with the costs that are associated when people are failing on a drug, right? So it costs money when people are sick. And so we are just as committed to showcase that our science works, to also untangle sort of the economics of it as well.
Perfect. Yeah, that sounds really great and very, very important, as you said. You need to show with the real data that actually can improve outcomes and bring that economic benefit to the healthcare system as well. I think that's the way to convince all the stakeholders.
Yeah, I'm a big fan of, like, I often say, like, hashtag show me the data. Like, I think they like the idea and are certainly have embraced like what we're up to. And now we're in the stage of getting that data so that we can make this a reality.
Perfect. And speaking of data, actually, I think as scientists, we try to be very rigorous about any sort of analysis that we do. Perhaps not so much so people do it in other fields. But what parts of your scientific training do you find the most helpful now for your entrepreneurial journey, perhaps outside of actual knowledge of the field and the technology that you are building? What helped you to become a successful entrepreneur that you learned in science?
I think determination. So any scientist, who's really worked in the lab knows very well that experiments don't always work. In fact, I think when you are doing cutting edge research and you don't have all the answers and you're in that discovery phase, I think, often most of the time you're wrong, right? Because you're still sort of figuring it out. But science taught me, to never give up, right? To throw myself into a material. And even when I thought I figured it out and then, something didn't work the way I expect, to not be discouraged by that, but to go back, and to push those learnings forward and, keep moving towards bringing forth, technology that I truly believe, can change the world. And, I really think entrepreneurship is very, very similar. You have to be determined. Olaris just turned 6 on June 1st. And, I'm a very reflective person. I keep a diary, and look back at, just what was going on, in those early days and throughout Olaris's journey. And at every point or at different points, people always told me no. And people told me, the technology was too early, or I was too young, or I didn't fit some mold, or the tech wasn't there, or there wasn't enough data, or there was a question about everything and anything. But if you remain determined, do the work, listen to the feedback, go back, iterate, learn the material, you can, push all that forward to create something and take just an idea and have it turn into sort of a bona fide product and a bona fide company.
Yeah, absolutely. I think that determination is the key. And as you said, in science, stuff does not work most of the times. Yeah. Actually, if an experiment works, that's kind of like a holiday. It's a celebration.
Have you ever run a Western blot?
Many times.
Like, I think there's nothing more frustrating in the world, or like that teaches you more like patience and rigorous than like a Western blot. I know there's new Lycor technology and things that have made it better, but like the old school, Western blot film that takes like 3 days to do, and that you like when you get those clear bands on a gel, like nothing is more satisfying than that,
Yeah, or maybe you realize in the middle of the process that you forgot to put the sponge somewhere in between the membranes and the gel, and and that happens all the time. Yeah, I think Western blot is like a perfect technique for somebody not to get into molecular biology, but actually to practice patience and determination.
Yeah.
Absolutely. And congrats, of course, on the birthday of Olaris. As you mentioned, you turned 6, so that's a big date. I think, especially for biotech companies, that's a very serious age. So congrats on that.
Thank you. I'm excited.
Perfect. And I know that apart from founding your own company, you are also very involved in different initiatives that support biotech entrepreneurship in general. And I would like to ask you, what are the common challenges that you see those biotech startups are facing these days, and how can those young founders omit some caveats that they might encounter on their way?
Yeah, I get asked this a lot, and it's kind of sad that like my answer hasn't really changed over the 6 years. So I think a huge challenge for entrepreneurs is access to capital, especially for females and people of color. I think, despite statistics that showcase, for example, that female-led companies actually have higher returns, unfortunately, only a fraction of funding goes to females, and I think even less so to minorities. Because, I don't know why, but I think that's a shame, because I think, good ideas are certainly not bound by race or gender. But unfortunately, access to capital still is. So, that's something that I think that we need to change. And I think there are initiatives out there. And so, one I think good example is this program that Massachusetts has called the Mass Next Gen Award, which I was lucky and fortunate enough to receive, where it recognizes sort of the next generation of female leaders in biotech. And this award gave me both recognition, which is, somewhat important, but it actually also gave me capital, which is a huge thing. It let me actually hire my first full-time employee, which then just let our productivity increase and opened me up to be out of the lab and doing kind of other things and sort of grow. And it also, importantly, gave me access to a really meaningful network of mentors that have been critical for Olaris's growth. And so, I think the challenge and the reality is that it is tough to get funding, especially if you're a female, especially if you're, a person of color. And honestly, if you're a young scientist, it's not easy in general, right? Even if you don't fit one of those other two categories. But fortunately, there are programs out there like the Mass Next Gen Award that help sort of bridge some of those gaps. And, as an entrepreneur, there are not enough hours in the day to get done everything that you need to get done. And there's lots of, different people begging for some of your time. I encourage people to look at programs like the Mass Next Gen or others that provide you with access to these things that are just honestly hard to get on their own, right? Like capital, like resources, networking, and things like that. And so, I think sometimes when we're as entrepreneurs, especially scientist entrepreneurs, we just think if we just develop the product, or we get the tech perfect, then everything will go from there. And that's not the case, actually. There's a lot more that goes into it to take a good idea and turn it into a company.
Yeah, I think there are lots of challenges, of course, and as you mentioned, funding, securing that capital is one of the most difficult ones, especially in biotech, because you actually need a lot of money. And what you just said, unfortunately, it's still the case that for women, people of color, it is still even more difficult to raise funds for the idea, and I hope that this is going to change in the next years. And I know that there are lots of initiatives who are trying to push for that change, and let's hope that this will be the case in the very near future. And you mentioned this Mass Next Gen Award, and in general, Massachusetts and the Boston area. It seems like it's a very fruitful ground for biotech companies, despite all the challenges. I know that you started in Harvard, but still I want to ask you, why actually Boston? Why did you decide to stay there and found your company there, not to go to Bay Area or somewhere else, but actually do your startup in Boston?
Yeah, I feel so lucky to be a native Bostonian and to be working in biotech, because I don't think there's a better place on earth to be a biotech entrepreneur. And then I am incredibly lucky and grateful that my family is here as well. Boston, I think, is arguably, one of the biotech or the biotech hub of the universe. It's got this sweet conglomeration of academia, hospitals, industry that all come together, that, are the people and the companies and the places or the organizations that are, really developing the solutions that are changing the world. Moderna, for example, when I was doing my PhD, Derek Rossi and Tim Springer, some of the scientific founders, those were, the guys that I went to beer hour with and had pizza with. And now, Moderna and Kendall Square is right in the heart of, hopefully coming to help the COVID vaccine and to move things forward. And so, Boston is just this rich epicenter where things can happen. And I truly believe if you can see it, you can be it. And so I think I'm a product of Boston, that from a young age, I saw what amazing cancer care was afforded to my brother. And then I was able to sort of jump in and work with, some of the who's who of top scientists to develop my own skills. and then, see and meet people that have started, companies. I've been fortunate, to be mentored by some amazing folks. John Abele was the guy that started Boston Scientific. He's, one of Olaris's investors, and he goes out for lunch with me usually, or actually I should say he goes out for breakfast. He's usually invites me 7 a.m. breakfast, which I'm like, John, really? But it's fine. I can get up that early. And he just passes on all this information. And so Boston is just this amazing place where, again, if you're willing to do the work, there's opportunities there.
Perfect. Yeah, it's great to hear. I've been lucky enough to be once in Boston.
Oh, you have to come back.
Yeah. Yes, I'll try to pay it a visit. Now it's kind of a little bit challenging, I guess, but hopefully it's going to change in the near future.
Yeah.
Great. Liz, before I let you go, I really want to take an outlook into the future together with you. I think personalized medicine is a very, very exciting field to be in right now. And I would like to take your insights into the future. What do you expect to happen in the field of personalized medicine in the next 10 years? Perhaps 3 developments that you see happening, or at least that you would like to see happening in the next 10 years.
Okay. Yeah. So I think, I'm by nature a very optimistic person. I think all entrepreneurs have to be, but I really think that we're going to see the things change in the next 10 years, that medicine will be very, very different. And so I think I'll think about it on both a technology side, a policy side, and even an educational front. On the technology side, biomarkers of response will be integrated into standard of care. I think we will, without a doubt, move away from sort of this current sort of guesswork that's associated with a lot of medicine, or, even worse, that you get the drug that your insurance company has negotiated the best price for. Biomarkers of response, like Olaris's and others, I think are going to be integrated into normal practice so that patients get the medicine that they think that they're getting and actually that they truly deserve. So I think we will see those without a doubt. On the policy side, I think that we're going to need to look at, what I and other people call more efforts for real world evidence. So we are fortunate to live in a time and an era where we have access to groundbreaking technology, and we can, use that tech to develop new products that have the potential, to impact patient care. This has led to a series of, what should we call this, drugs or treatments that come about that go through what's an accelerated approval process, where companies and manufacturers can apply for what's called breakthrough status, that they have, a drug and early data, it shows such promising sort of output or outcomes based on some surrogate endpoints that regulatory bodies like the FDA fast track it so it can get approval and get to patients, right? Because getting access to drugs is super important, and we certainly don't want to slow that down. But unfortunately, oftentimes sort of the larger studies that are needed to truly understand how drugs work in a larger patient population is not done. And so I think we need to kind of commit to what's often called real world evidence that really tries to unpack outside of a very controlled and curated clinical setting, how do drugs really work? And what is the value that you can associate with that? Some drugs now, there are drugs that have $1 million price tag, right? And they may or may not, extend life or do something for weeks to months. And, I'm not one to assign what is the value of those time points, because I think actually for different people, it has different value. But I do think we need to commit to doing those larger term studies so that, again, all the stakeholders involved can have a better understanding of how things actually work and they don't. And that's the third thing that I think we'll see sort of change is education. On the education side, I think we have access to more data than ever before and information ever before, but medicine and health is still sort of, fragmented and almost like a little bit controlled. And I think that in the future, as we develop more tools and more platforms, we're going to see patients, and even not patients, just individuals, take more of an active role in kind of controlling and building a healthier future sort of for themselves. And so, I think part of these things are actually probably all connected, biomarkers of response, real world evidence policy, and education, that collectively these things, and I'm sure others, will come together to transform how medicine is 10 years from now.
Yeah, that sounds very exciting, and it sounds like a great future to expect and to live in, hopefully in 10 years. I guess that's where we all want to go, and I'm pretty sure where we'll all be in 10 years from now. So, Liz, thank you very, very much for this interview. Now, very last question before I let you go. I know that there are a lot of people in our audience who would like to reach out to you. How can they do it, and where can they find you online?
Awesome. Yeah. Well, again, thank you for having me. And I would love to follow up with people. So welcome to reach out. I'd say LinkedIn, Facebook. You can even, stalk me on Instagram if you want, or simply through our website. And you can send an email at info@olarisbor.com.
Perfect. Liz, thank you so much for this very, very exciting interview. It was fun talking to you. I learned a ton, and you really made me want to buy an X-Men comic and the next ticket to Boston.
I think that sounds like a plan. I like it. Thank you so much.
Perfect. Yeah. Thanks a lot. And I'm looking forward to speaking to you soon again in the future.
Awesome. Thank you.
Thank you so much for being with us today on the Personalized Medicine podcast. If you like this show and know someone who would enjoy it too, please share this podcast with them. And don't miss the next episode yourself. Subscribe to the Personalized Medicine podcast on your favorite podcasting app. You can find us on Apple Podcasts, Spotify, Pocket Casts, Stitcher, iHeartRadio, and many, many more. Please rate us there and leave a comment. That helps us to grow and deliver the best experience to you. To access the show notes for this episode, visit our website, pmedcast.com. It's pmedcast.com. And engage with us on social media, where we regularly share the news and exciting content on personalized medicine. You can find us on Twitter, LinkedIn, and Facebook just by typing in Personalized Medicine Podcast, or use our handle, pmedcast. And if you have any feedback or would like to suggest a guest for the show, write us an email to team@pmedcast.com. Have a great day, and until next time.